Well, if if all delegates are ready, I think we can take our seats so we can start. If all delegates are ready, I think we can take our seats so we can start. If all delegates are ready, I think we can take our seats so we can start. So, good afternoon to all, and thank you for your participation, and welcome to the University of Calabria. I'm very pleased on behalf of the World Organizing Committee to welcome you to the 23rd edition of the European Erasmus Branch Association meeting. Today, a great dream is fulfilled for me, because it was when I was a student that my dream was to accomplish this very important scientific event here in my university and in my land, and today this dream has come true. For sessions, after I will stress with more emails, I think you will not applaud me anymore. For sessions, 2 workshops, 3 keynote speakers, 71 oral presentations, 59 posters, 205 delegates from 30 countries inside and outside Europe. And I hope that all we together will contribute to achieve our goal, that is to communicate Erasmus branches. Obviously, all this would not have been possible without the support and help of many people, but in particular, all my young collaborators who have worked tirelessly in organizing this conference with me. And now I will show you a short video, 1 minute, to summarize the lines of research on sharks in our group. Just 1 minute. The lines of research on sharks in our group. Just 1 minute. Thank you. As you know, the president of our scientific committee is Leonard Compagno. We collaborate with him for many years, and we wanted with all our heart that he was here today. Also because one of his grandparents was. Unfortunately, his health conditions not allow him to be here, but in this moment he is following us in streaming, and please, I beg you to dedicate him a round of applause. Hi, Leonard! And Leonard recorded a video message of few minutes for us, and it is with pleasure that I ask you to listen to him. A video message of few minutes for us, and it is with pleasure that I ask you to listen to him.
Well, even though I'm not physically there, I am certainly electronically there. But so all you people that have come to the Shark Conference, I welcome you with all my heart and mind, and like in the Shark Conference, I welcome you. Sharks are not a mined out field when it comes to their study. In fact, the more we find out, the more we realize how much we don't know. And in terms of diversity, in terms of their behavior, in terms of many other things. Well, the country Italy means something to me because I'm partially Italian. Eugenie Clark, in a 2011 edition of her book *Lady and the Sharks*, wrote something on the copy she gave me while I was in Stockholm, and that is this. To Leonard, the greatest shark expert on the planet. I'm not so sure I can live up to that, but I'll try sometimes. I think the first paper I ever wrote was something back in 1967, I believe, on tooth pattern reversal in sharks. In other words, sharks have a certain pattern of their teeth, and all of a sudden it reverses itself, and I discovered that, and it hadn't been really discovered before, and from that I went on to just about anything that interested me. I've published quite a few things. I'm not so sure how many exact number of papers, but it's upward of 1,000. I've worked on all groups of sharks, and I've worked on them globally. So I've had an effect on their systematics or taxonomy and general biology worldwide. One of the things that was a particularly good paper is that one time I met a Hawaiian friend who's also a shark researcher, Leighton Taylor, and he showed me some material from a giant new species of shark that had been collected off of the Hawaiian Islands, that is the megamalt shark. And so from that, essentially, I wound up describing the thing, and Leighton and another friend, Paul Strusaker, added bits and pieces to it, but essentially it was my job. What I'm busy working on now is a grand classification of the shark-like fishes. I've come to a plan that apparently they represent two major groups, the chimeras and the placoderms, and the acanthodians or spiny shark, and the sharks proper. And I need a bit of work to do that, but it's a little bit different from what people have done before. My involvement with the Jaws movie was serving as their chief scientific collaborator. Well, about Jaws, it had a dual function. For one thing, it tended to mean that people had a tendency to kill more sharks, but then again, what it had a tendency to mean is that people got more interested in sharks and got more conservation-oriented to sharks. On the importance of shark research to communication is that it's vital, that it paraphrases what Albert Einstein said one time about research. If you confine research to the purview of researchers, it doesn't get out to the rest of the world, which is unfortunate. It should get out to the rest of the world. Well, I think there's a problem of the opinion that people have with sharks uh, besides, they do it from the standpoint of not really knowing what sharks are. There are a lot of different species of them, and the average size is about that long, and which can't be particularly harmful to anybody. And even with the ones that are big enough to bite you, they are for the most part. They don't do that at all. In fact, if they did do that, I wouldn't be here. My advice to would-be shark researchers is to do it, and because sooner or later your opinions will become unique. Your opinions will become something that is adding new knowledge to our field. Well, what advice I would give to other scientists is that the game isn't over, not by a long shot, and there's a lot more to learn out there. If you're studying sharks, keep studying sharks. If you're not studying sharks, you probably should be studying sharks. I wish for all of you delegates to the meeting all the success in the world in your studies, past, present, and future. I wish for all of you delegates to the meeting all the success in the world in your studies, past, present, and future.
Now I introduce the rector of our university, and I thank him for having provided us with the structure that hosts us, and I invite him to extend his greetings. Provided us with the structure that hosts us.
Just look fantastic, the video. The shark, very soft music. Very, very nice, complimentary, very perfect combination. Now I give you welcome to Calabria, and welcome to University of Calabria. I am a geologist. I am to Calabria, Professor Speroni. I'm very happy to give welcome to university, because the Calabria University is a very strange university in Italy. Because usually in Italy, the university, the department is in different place in the city. Distance between two departments usually is kilometers. In this university, distance for department is just a meter. Because the University of Calabria is the really American campus. We have a lot of things. All departments are together. We have a lot of possibility to give apartment for students. We have 2,500 apartments now for the student. I stop I just show you one video, give idea about this university.
The University of Calabria is the most equipped campus in Italy. The university is located in Calabria, among the hills of the University Valley, between the towns of Cosenza and Rende. It is surrounded by the uplands of the Sila and the Pollino, and its campus lays on a green area of over 200 hectares. Its surroundings and architecture are the hallmarks of the campus, which was designed and developed by architect Vittorio Gregotti. The long bridge that runs across the whole university was built among the hills of Arcavacata. The bridge is connected to the department buildings, commonly called cubes, hosting classrooms, laboratories, and all the other facilities and offices. With 79 degree courses, 3 year, master's and single-cycle degree courses, interfaculty masters and second-level degrees, and over 14 departments, the university provides higher education opportunities and specialization thanks to more than 22 postgraduate courses. "30,000 students populate the campus, and over 800 of them come from foreign countries. UNICO provides a multicultural approach. Foreign students attend courses, participate in conferences and laboratories organized across the whole academic year, owing to the memorandums of understanding signed with other universities at an international level and thanks to the support of the University Language Center. The University of Calabria supports the students' right to receive education with over 6,000 scholarships, and it fosters international students' mobility. The residential center offers students 2,500 bed places in the various university accommodation units scattered across the campus. Living on a campus means taking courses in over 300 equipped rooms and labs. In the largest library system of the south of Italy, with about 400,000 books and several opportunities of online accesses. Doing lots of sport activities at the University Sports Center that offers 25 different disciplines and several sport facilities. Observing seasonal alternation of the various plants at the Botanical Garden, or learning more from the Museum of Paleontology, Zoology, Natural History and Environment. Going to theatrical performances or to the cinema, and in order to learn more, participating in concerts and in the various activities organized by the Center for Arts, Music and Theatre. On a daily basis, the 5 university canteens host 1,300 students and provide 3,500 meals. The campus also hosts a health center, a bank, a post office, and a kindergarten. Disabilities are not a hindrance for students at Unical, thanks to the support provided to students with learning disabilities and special educational needs by the Service Center. Unical meets the requirements of all students. The Guidance Office offers support to students since the very beginning of their university experience and when entering the world of work, thanks to meetings held with companies that operate in the territory. The University of Calabria helps students in transforming ideas into innovative enterprises with the support of the Liaison Office and the Technist Incubator, hosting startups and spin-offs generated by the research carried out in labs and excellence centers. It is also the most sustainable and environmentally friendly campus in Italy due to the numerous energy systems in an advanced stage of design: geothermal, plain solar, concentration solar, LED and PV. Residential areas, education, innovation, multiculturalism, sustainability. These are all the features that make the University of Calabria a reference point for sustainability, as it is demonstrated by international rankings and awards received by the university. The University of Calabria is a real excellence within the university system. It is your right choice, an opportunity not to be missed. What are you waiting for? University system. It is your right choice, an opportunity not to be missed. What are you waiting for?
Okay, this video gives exact idea about the Calabria University. Okay, I hope you have time to visit the university you visit Cosenza, very historical city, and you visit Calabria. Because around the world, the name Calabria is no very good name, but it's not true. Calabria is a fantastic region. Same environment is very nice. For me, Calabria, I have a one big problem. That the food is very good. It's very difficult to make a diet in Calabria. Allora, thanks, good work. See you again tonight, tomorrow, because when the Speroni invited me to live and to eat, I absolutely I didn't
Also
say
because we will have good food and good wine above all.
no. Good wine.
And now Professor Cesare Indiveri, that is the director of our department.
I would like just to welcome everybody. I have no video to show you. Just I would say that I'm very happy to see so much scientists working on the topic of sharks. I'm surprised because I have a personal opinion. In spite of my research topic, I do research on biomedical sciences. Everybody is convinced that humanity will be saved by biomedical research. I am convinced that it is not true, because humanity is in a large system, environment, so only the knowledge of the whole environment will save humanity from destroying. So for this I am very happy to see you, and I wish you all the best for the meeting, and ad maiora. Thank you very much.
Many thanks. Last but not least, our the president of the EEA, Alex Bartoli.
Thank you very much, Emilio. I will be very brief, because it's not important what I have to say, but which is really important is what you have to say with your presentation and what is why we are all here. Thank you everybody for coming. Amazing numbers, what you said. So congratulations for the organization. And I have to say that it's real that it was his dream, because it was one of the applications to organize the EEA with most anticipation and with most worry and with concern. So very well done, and well job, Emilio, and all the team that has made this possible. So thank you everybody for coming. Thank you to all the organizers, Greece, the Centro della, and of course the University of Calabria for hosting us, and all the colleagues and volunteers that have been hardly worked to make it possible. Very excited to see all the talks and all the posters and see how the science on sharks is evolving, because it's very important, first for the knowledge itself, but also because it's vital and very important for management issues and policy issues, which is very important in times where sharks are, or some species are very depleted. Everything, just to let you know that this year are coming some interesting topics on policy issues, as we were discussing this morning in the board meeting. Mainly two focus of importance that all of science that we are doing here could help. One of them is trying to reach the zero catches for the mako sharks in the Atlantic, because the populations are really depleted. So we'll see what happens in the future. And then again, we will start this kind of sort of a fight on the fins naturally attached regulation, because the reviewing is coming, and we will fight for keep this regulation. Well, that's all. Just in terms of institutional things, remember, for those who can be interested, that at the end of the talks today will be the General Assembly here. So who wants to stay, it's very welcome to stay to the General Assembly. It will be from 7 to 7:45. So it's just 45 minutes for those who can be interested, and that's all. Let's start.
Well, I need,
Let's start.
because before starting work, I want to tell you that this summer our young colleague Zeddy Seymour lost his life in an accident at sea. Zeddy was one of the first to contact me for the meeting last spring, and for this reason I want to invite Francesco Garzon from Mara Alliance just for saying few words in his memory. Please, Francesco. Francesco Garzon from Mara Alliance just for saying few words in his memory. Please, Francesco.
I'm not quite sure what to say, so I'm just going to keep it brief. If any of you have met Zeddy before or knew him from conferences, you know he was a great person and a very passionate conservationist. He really loved sharks and was really dedicated to doing something to change the situation for them, especially in Cape Verde, where he set up the Mar Alliance project, and he had been working for several years. And he did manage to do something very nice there that we hope will manage to keep going. So rather than saying much more, we'll show you a brief video of him explaining what he liked to do and why he thought it was important to do it, and we thought that would be the best way to remember him, really. So why he thought it was remember him, really.
Estamos aqui no área de berçário, aqui na Boa Vista, e estamos fazendo uma pesquisa científica utilizando a pesca científica. Então, como eu disse, essa área é muito importante para os juvenis e os neonatos, os bebês de tubarão, de várias espécies. Confirmamos 6 espécies de tubarão e uma espécie de raia nesta baía. E então agora estamos a tentar capturar alguns indivíduos para poder fazer medidas, tirar amostras e até a marcação, com o objetivo de identificar o quão importante é esta área durante esta fase de vida destes tubarões, quantos meses ou anos ficam aqui, em quais zonas e habitats. Aqui na Boa Vista, tanto como no mundo, a ameaça maior é a sobrepesca. Mas junto com isso, a falta de conhecimento e sensibilização do tubarão, obviamente existe a percepção que o tubarão é má. E esta ideia, na verdade, é tão errado, porque o tubarão não é este monstro que a gente pensa desde criança até adulto, e muitas pessoas ainda continuam com essa percepção, mas o tubarão é uma parte muito importante do ecossistema e mantém a saúde dos ecossistemas, das populações de peixe e até do mar. Sempre trabalhando com os pescadores artesanais, porque são eles que estão no mar dia a dia e são eles que têm esse conhecimento incrível, tanto dos tubarões e as outras espécies, quanto do mar e os habitats que tem aqui na Boa Vista. Então é com eles que a gente trabalha também na educação, a capacitação e a sensibilização, mas igualmente com as crianças nas escolas. É com eles que a gente trabalha da comunidade para tentar melhorar essa percepção do tubarão e tentar salvaguardar as populações que ainda tem aqui em Cabo Verde.
Okay, now we are coming to start, and I please invite you to respect the times, also because As you so we have a lot of thoughts. So I invite the chairs of the session of biology and physiology to come here, and so we are going to start. And I wish all of you good work, and maybe before the coffee break, I will have some information for you. And good work to all. And maybe before the coffee break, I will have some information for you. And good work to all. And maybe before the coffee break, I will have some information for you. And good work to all.
Okay. Good afternoon, everybody, and welcome to the first session of this meeting, the session on biology and physiology of elasmo branch. I am very glad to be here, and I'm sure it will be a very interesting session. Only I wish to remind you that the keynote and the invited speakers will have 25 minutes for their talk and 5 minutes for discussion, while delegates will have 10 minutes for the talk and 3 minutes for discussion. Okay, we can start with the first speaker, an invited speaker, which will be introduced by Bruno Tota, Prof. Emeritus of Physiology of the Department of Biology, Ecology and Art Science of this university. Thank you, Bruno. Thank you all, and enjoy this session.
Thank you very much, Professor Edwin Taylor. London, during the heavy bombing of the city by the Nazi attack, his basic first experience in physiology was Southampton, where there were two masters of the field, Professor Graham Shelton and Professor Gerald Kirkwood, that maybe is well known because he was a long-run editor of comparative biochemistry and physiology. After this first stage, Professor Taylor was invited by Professor Lowenstein to move to Birmingham at the physiology department, where he had he made all his career until he retired in 2006, and then he became Professor Emeritus of Physiology. The interest of Professor Taylor were encompassing acid-base equilibrium in crustacean and mollusks, with a particular reference on how, for instance, lactate could modify the hemocyanin oxygen binding curve. His main research moved to elasmobranch species, and he studied the control of the respiration cardiac cycles using electrophysiological and pharmacological and then on hemodynamic methods. One aspect of the research which may be historically important for this university was the involvement of Professor Taylor in two European projects, one on the relation between thermal development and thermal heating and ill development in North Italy, and another one on the importance of polyunsaturated fatty acid on sturgeon performance. These two projects were in cooperation with Master of Physiology like Professor David Randall, and with us, with Claudia Nisola, Professor Nisola in Naples, and some of the people of the physiology group that now are professors. Professor Taylor was not only teaching and making experiments in Birmingham, but he was almost everywhere. So you could find him many times in Canada, in Brazil, in Italy, you could find him many places. This emphasizes his international oriented mind and character. And it's also for this that, of course, he liked to be with us sometime. After his retirement, he is spending most of his research time in Brazil, where he dedicated himself to the respiratory, the control of respiration and cardiac cycle, particularly in reptiles. Please, go to the podium. Please, go to the podium.
The title of the talk of Prof. Taylor is Cardiorespiratory Integration in the Brainstem of the Dogfish Sciliorinus canicula, a neuroanatomical and neurophysiological study.
Can you hear me okay? Yep. I've cut my slides to a very low level, and I will speak slowly because I'm aware that speaking in English the way I do is difficult. I would like to start by thanking Bruno, a very old friend of mine, for his introduction and for causing me to be invited to this meeting. We've known each other many years, 30 or 40 years, and have worked together, as he's told you. I'd also like to thank the two chairpersons, Sandra and Maria Camella, who again are old friends of mine from those days, and also Maria das Graças Sarjo, who's looking after me on the trip. Okay, so the first slide is showing where I've worked for 42 years. The University of Birmingham is in the centre of England, and as Bruno told you, although I worked a lot of the time there, I also worked in marine stations and research institutes all over the world, basically. And what I'd like to talk to you today about is what the work I did on elasmobranchs in... Could you stay in the slide? And what I'd like to talk to you today... Okay, thanks. Whoop. I think I better do it. Okay. And the animal we chose to work on is this one, Sciliorinus canicula. I notice there's a T-shirt that features this animal. The wonderful thing about it is that, well, we've called it a model. I'm actually against the idea that animals are models, but I'm emphasizing the fact that it's an amazing animal for looking at the evolution of cardiovascular control, for reasons I'll try to illustrate, okay? This, the dogfish, and I apologize because many of you are ecologists and conservationists, so you must hate the sight of a dissected dogfish, but these are cat sharks, they're called cat sharks these days. These are museum preparations. When I was a high school student, when I was only 16, we were shown this sort of thing and had to do our own dissection. This is dissection from the ventral side of the dogfish, showing the heart, the ventral aorta. On this side of the animal, the afferent branchial arteries to the gills. On this side, the efferent arteries collecting that blood and distributing it along the dorsal aorta to the fish. Okay, and you're well aware of the circulation structure of a fish, which is singular. This is a dissection from the dorsal side of much more interest to me, showing that the animal has a relatively large brain. I'm sorry for the shaking. And so this is the brain. The spinal cord is deflected to the side here. The great thing about fish is that all the respiratory nerves and the cardiac nerve are concentrated around this area and project into the brainstem. So it's a wonderful preparation. Any fish is a wonderful preparation to look at respiratory cardiovascular coordination. But it's particularly true of elasmobranch fish for reasons I've tried to summarize here. Now, this is important to you conservationists. I worked on these animals back in the 70s, 80s and 90s. In those days, at least, the animals were very common in the wild around the British coast, and they were caught from the wild and taken to the Plymouth Marine Lab, some of you may have heard of, down in the west of England. And we did work on these animals caught from the wild. I'm basically opposed to the idea of killing animals from the wild for experiments, but this was necessary. As you know, they're an ancient group. The great thing for an experimentalist is they survive well in captivity and they make excellent preparations. They're very tough. We decerebrated them after an anesthesia, of course, decerebrated them and often spinalectalized them as well. We could cannulate vessels, the ones you saw just now. We could expose peripheral nerves and the central nervous system for recording and stimulating, okay? All the respiratory muscles are inserted around the head and pharynx. As I said, the respiratory nerves are the 5th, 7th, 9th and 10th cranial nerves, and the 10th cranial nerve also supplies the heart. But the elasmobranchs are unique in that they only have the single innervation of the heart. They do have a sympathetic nervous system. Are unique in that they only have the pharynx area, so that the heart only has a parasympathetic innervation. So if you want to study the role of the vagus, by the way, vagal innervation is predominant in control of resting control of the heart or chronic control of the heart throughout the vertebrates all the way to mammals. But if you want to study that control, it's a good idea to work on an animal which has only that supply. So this makes this a unique preparation. And this is a diagram of what I've just been telling you. This is the 5th, 7th, 9th and 10th cranial nerve. The 10th cranial nerve innervates the four rearmost gill arches. The first one is innervated by the 9th cranial nerve. There is a significance to that, which I don't have time to talk about. The spiracle, which is an inhalant passageway, as well as the mouth, of course, is innervated by the 7th cranial nerve. The mouth, of course, by the 5th, which innervates the jaw muscles, okay? So these are the respiratory nerves. The next diagram, which is and these, of course, are the centers in the brainstem, from which the activity you trace in these nerves originates, and this is the sensory projection, okay? The reticular formation here, although it's uncertain in fish, is the probable site of respiratory rhythm generation, but that's another story, okay? Now, looking at this from the dorsal view, which is the kind of diagram we worked with, this is the 5th cranial nerve, the 7th, the 9th, and the branches of the 10th innervating the gills, but there are branches of the 10th cranial nerve innervating the heart, okay, running down the ductus cuvieri. One branch from the last branchial branch of the vagus to this last gill arch. There's a small branch that goes down the cuvarian duct, innervates the heart. There's also another branch from the visceral vagus, which goes to the rest of the. You know that the vagus nerve innervates the liver, the gut, and many other organs in the viscera. It's called the vagus nerve from vagare, Latin vagare, which means the wandering nerve. So there's another branch to the heart. So there are 2 branches from the vagus to the heart, and we'll come back to those if I have time. So this anatomy was to expose nerves for recording and stimulation, and also inject nerves with neural tracers so we could look at their projections into the brainstem. The types of neural tracer we used were very primitive systems like the cobalt. Cobalt's really good because it traces the axon tracts as well as the neurons themselves. This is the 4th ventricle, the space, not to be confused. The heart, of course, this is a ventricle, is a space in the brainstem. The 4th ventricle is in the medulla oblongata, and there's a bunch of nerves in this nerve cells in this area, the dorsal vagal motor nucleus, which is a predominant area for respiratory neurons and also for cardiac neurons, and then dendrites project ventrolaterally from these nerves. So we used simple systems like this. We also used a rather complex system to use called horseradish peroxidase. This shows the sensory area of the vagus projecting to the side here, dorsal to the motor nucleus. This is sensory and motor, again next to the fourth ventricle. This is a section, of course, of the brain stem. So having put the traces in, you kill the animal some time later, depending on the migration routes that you have to calculate, and you then kill the animals and section the brain. Now, what we did was plot the distribution of the groups of cells innervating the 5th, the 7th, the 9th, and the branches of the 10th, supplying the respiratory muscles, and also the 2 branches supplying the heart. And you can see they're like a ladder. They project back through the brainstem. So the organization of these groups of neurons innervating these motor cells are sequential through the brainstem. And that's interesting because it may be thought of as primitive, not simple, but primitive. And what we did then, of course, was record from these nerves and discover that they fire sequentially. So this is the 5th mandibular, and this is the 9th glossopharyngeal. And you can see that if you look at that burst of activity we're recording from this nerve, so this is a respiratory burst. Each time that burst happens, then the mandibular muscle innervating the jaw is contracting in the normal fish. And this is the glossopharyngeal, okay? The 9th cranial nerve innervating the spiracle, and the spiracle's muscles will, of course, be caused to... You see they're out of phase. And then if you look at the phase for... So this is the 5th mandibular, the 9th, the 1st branchial, and the 4th branchial, and this is actually the cardiac branch. You can see the cardiac branch is a noisy signal. That's not a problem with recording. That's because there's more activity going on in this nerve than there are in these other nerves. They're showing clear respiratory activity. These are, of course, basically they're integrated recordings, so they're not spike counting basically. So that's all the action potentials there counted and plotted as a curve in that way. So, and this is the one of the gill arches contracting just to show its respiratory activity. So that's a recording of the bunch of nerves that I've been talking about. One of the gill arches. Now, the reason that we're interested in this is because I know this isn't, I apologise, it's not an elasma brain, it's a textbook diagram of countercurrent exchange. You know that water flows through the gills of fish, and then through the gill lamellae, and the blood runs in the opposite direction. This is a countercurrent gas exchanger. It's highly effective. We don't have time to talk about respiratory gas exchange, sadly. I've got to get on with it. But this is the picture of shark gills. This is a diagram of this situation. There's water flowing in one direction, blood in the other direction. This is what the gill lamellae actually look like, and these are pillar cells celebrating the blood channel. So blood's flowing continuously in one direction and water in the other, and they flow at a rate which is related to the gas conductance of the 2 media. So blood, because of hemoglobin in red cells, conducts much more oxygen than water, so you get a much higher flow of water than blood, but these are matched according to their capacities. Now that's interesting because both of these flows are pulsatile. Water than blood, but these are matched directly to the gills, so blood flow through the gills is very pulsatile, and so is water flow. One would therefore postulate, or hypothesize if you like, that the 2 flows would be coordinated, and in fact they are under certain conditions. There's a big debate still about whether these things actually happen, but cardiorespiratory synchrony certainly can happen, and that we're just studying the mechanisms by which this can happen. So this is now recordings from a cardiac nerve innervating the heart, and the branchial nerve innervating gill arch 4, which is branchial 3 from the vagus. So they're both vagal branches. This is a respiratory branch, this is the cardiac branch, and this is a decerebrated animal, okay? And it's force ventilated, also paralyzed, so it's force ventilated. So there's no rhythmic water flow here. So this is feed-forward activity. In other words, the brain, the respiratory center in the brain, is generating activity in the respiratory nerve, and extraordinarily, the same activity is present in the cardiac nerve. So the heart is receiving information about ventilation. Hope you understand that. This is a few fiber recording, splitting fibers down. You can see individual fibers here, and you can see there are some fibers which have rather large action potentials, which show respiration-related activity. And then there's other activity which isn't respiration-related totally, which is smaller fibers. And you can see that the activity in these fibers increased during hypoxia, which tells you they may have a different role. In this case, it's hypoxic bradycardia. Now, to check this out, we tried capsaicin, which is what's in red hot chili peppers. This stimulates something called J receptors in mammals, and if you do this with a dogfish, a cat shark, when you inject the capsaicin here, something very interesting happens. There's a huge increase in respiration rate. So this is a branchial nerve to the respiratory muscles, and the firing rate increases. But the branchial cardiac branch, the branch to the heart, shows a similar increase in this activity, but an enormous increase in the background activity. And that huge increase in activity actually causes the heart to stop. So the animal gets this nasty toxic chemical in its inhalant water and stops the heart and increases ventilation to get rid of that. And so this indicates that there are two separate roles for these two sites of activity. So this is some of our evidence. I'm only showing you glimpses of what we've done because I don't have much time. Now what we did then was detailed neuroanatomy, and this is injecting the tracer into the cardiac nerve. So all these cells here, these black things are cardiac neurons, are all innervating the heart. And it's only the heart that has this group of cells. This is the dorsal motor nucleus of the vagus, which also contains respiratory neurons. So these are cardiac neurons mixed up with respiratory neurons close to the fourth ventricle in the dorsal motor nucleus. But then there are these scattered neurons outside the dorsal motor nucleus. They only innervate the heart. They're uniquely cardiac cells. Now, in order to find out what they were doing, remember that there's two sorts of activity in the cardiac nerve. There's bursting activity, respiration-related activity, and the other activity which increased with capsaicin during hypoxia. So there are two sorts of activity. Now, there are therefore two groups of nerve cells, one close to the fourth ventricle, and the other scattered group of cells outside that area. What we did, and this is a difficult thing to do, and I can't describe the technique, I'm afraid, we then placed electrodes in the brain looking for these cells, and this is what we found. When we found a cell that was in the dorsal motor nucleus of the vagus, this is the activity in the cardiac nerve, this is the activity in that cell, and you can see that it's contributing activity to each of the bursts of activity. So it's respiration related. When we touched the gill arch, there was a burst in the nerve and a burst in this cell. So this cell is getting feed-forward information from the respiratory center in the brainstem, which causes it to be respiration related, and it also responds mechanically to mechanical stimuli of the gill arch. Okay? This cell, on the other hand, which is outside that area, is firing, but it's firing completely independently of the respiration activity. This is the nerve again, the cardiac nerve in the same animal. You can see a lot of activity going on with respiration-related stuff. This nerve, this cell, this single cell, is firing independently, continuously, but not with respiration-related activity. This cell is even more interesting in its own way because it was completely silent. The way we locate these cells is by stimulating the cardiac nerve back into the brain. It's called antidromic stimulation, and then looking for cells that fire. It's a little bit more complicated than that, but that's what we back into the brain. When we switch off the stimulator, it's completely silent. So it's not saying anything until you touch a gill arch. You touch a gill arch and it fires. So this cell is uniquely stimulated reflexly and not centrally. Okay? So these are three populations of cells innervating the heart in the brainstem. And this group here, because it's mixed up with respiratory neurons, not surprisingly showed continuously respiration-related activity in the brainstem. and they've got very fast conduction times. The axons, the motor axons leaving these cells have got very fast conduction times. This is necessary in order to cause beat-to-beat control of the heart. If you're going to very carefully control heart rate, you need to have immediate conduction, high conduction velocities for immediate contact, and this is the same reaction rate as conduction rate as B cells in mammals. So in fact, the stuff where I'm describing to you now in elasmobranchs is very similar to what I know. There was a group that I was well acquainted with and worked alongside in Birmingham that worked on mammalian cats, dogs and rabbits, and the sort of information we got from the dogfish was very similar. So it's an interesting evolutionary story. We're looking at the early stages of the evolution of this system. Okay. So this is a summary which I think I've more or less said all of that, so let's move on. Because we found that there was phasic activity which in these nerves, we wanted to find out if they could phasically affect the heart. So what we did was phasically stimulate the nerve. Now if we just stimulate, now this is the continuous, So this is, if you just stimulate, this is a messy, doesn't matter. This is the stimulus to the cardiac nerve, and you can see it slows the heart dramatically because the vagus is an inhibitory nerve. If you stimulate the nerve continuously but put on top of it, and we had a stimulator that could do this, phasic stimuli with a higher voltage, you'll see that it phase locked to that activity. So this looks a little bit like what we're recording from the cardiac nerve, background activity, but on top of it, larger units which were respiration related. And you can see the heart, because this is blood pressure, the heart locked to that rate. And if you do this on a whole bunch of rates, with the resting rate at 37, 36, you can slow the heart to 19, all the way through the 20s to 36, up above that rate, okay, all the way to 41. So in fact, these bursts of activity in what's thought to be an inhibitory nerve can actually drive the heart faster than its resting rate. In other words, the vagus has a much more sophisticated role in controlling heart rate and, of course, relating it to ventilation than is normally considered. The parasympathetic vagus is described as an inhibitory nerve. It isn't simply an inhibitory nerve. It can drive the heart at different rates, okay? If you stimulate a sensory nerve centrally, in other words, a nerve from the innervating a gill arch, you stimulate that with pulses. That also entrains the heart, which of course relates back to the fact that if you touch a gill septum, it affects heart rate, and this is mimicking that by electrical stimulation centrally towards the brain, and this also phase locks the heart. So it shows the heart can be phase locked by this activity, whether it's coming from central feed-forward control or reflexly from mechanoreceptors on the gills. Okay, so it's a complex system, as you'd expect, and just as complicated as any mammal. These animals are not simple. So that's another summary which we won't go back over. The important thing is that there are two, remember I told you there are two nerves either side of the fish, two different cardiac nerves, and we were interested in whether they had different roles. And if we stimulated them peripherally, we found that the branchial branch that came from that last branchial branch of the vagus had an enormous effect on heart rate. Stimulate that nerve peripherally towards the heart and the heart stops. Stimulate the other nerve peripherally and the heart does slow, but it doesn't stop. If you do the opposite thing with these two nerves, if you stimulate the branchial nerve centrally, it has practically no effect. Stimulate the visceral cardiac nerve, the one from the other part of the vagus, it stops the heart. This shows that this nerve is predominantly sensory, and this nerve is predominantly motor. Okay, so you've got two different cardiac nerves, one effectively a motor nerve and the other one effectively a sensory nerve. Now this means that this preparation is amazingly useful for studying cardiac control because you have two separate nerves playing separate roles. And I've tried to, I was asked to write a couple of chapters in the Encyclopedia of Physiology, which was published in 2011, and this is the diagram I put together. This is the shark heart and the ventral aorta, a diagrammatic version of the gills. This shows that and these are the centers in the brain, the sensory center, the motor center near the fourth ventricle, the ventrolateral group of cells, and these both innervate the heart, one generating respiration-related activity, the other one generating activity which can cause toxic bradycardia, as you remember, and then the other cardiac nerve is primarily sensory, projecting to the sensory area in the brain. Okay, and so that, and then this is the generation of respiration comes from this area here. And this is a diagram of the probable interactions in the brainstem. I have no time to go through all of these. It would take an hour, and you wouldn't want that. I'll be thrown off the stage, of course. And finally, quite rightly, finally, there's this rather nice diagram, which is a straightforward diagram. The heart, the gills, the feeding muscles. I've not talked about this at all, but sharks can, you know, the feeding muscles where they grab fish and swallow them can be recruited into respiration. I've not talked about that at all, but the central control of all of this we've thought about and hypothesized. And if you want to know the arguments, you should read this, which is a book published a couple of years ago in which we generate a Twitter chapter. And the diagram I've been boring you with today was very kindly by the publishers put on the front of the book. So, and there's I think that's a blue shark, and there's a beautiful shark. There's my crummy schoolboy diagram, and they've conveniently related them so that you can describe the two. So if you want really to know what I was talking about, buy the book and read the chapter. Okay, thank you. Thanks very much.
Okay, thank you. Very interesting presentation. You can open the discussion. Question, please. Thank you.
Holly.
Open the discussion.
Yep. You mean you don't always have cardiorespiratory synchrony? Yeah, okay. That's an interesting one. Well, sharks are complicated, just like any other animal. In our animals, we were able to show long periods of cardiorespiratory synchrony. In animals cannulated, because we didn't have remote sensors in those days, with long cannulae and left undisturbed in gigantic circular chambers full of fresh seawater. Under those circumstances, the animals settle, and particularly during the day, they're not particularly active, and you get one-to-one synchrony for hours. So I think they use it to optimize gas exchange and save, okay? When they stirred or swam, it disappeared because heart rate accelerated, and when a shark's really swimming fast, of course, it ram ventilates. So then the integration is unnecessary, and we've not studied this, and as far as I know, no one else has. But I would predict that when a shark accelerates to ram ventilate, its heart rate will increase. Now that sounds intuitive because they would want the heart to pump fast to provide oxygen, but it's not just that. It's because when the respiratory generator switches off, and the respiratory generator generates these pulses of activity to the heart. If they switch off, the heart's going to accelerate. So in a way, the mechanism is in the brainstem to cause the heart to accelerate during ram ventilation, okay? So there are lots of respiratory physiologists around who told me they don't think cardiorespiratory synchrony exists. I think it does. It's not a belief, it's to do with examining the evidence. And what we've done is uncover in detail the mechanisms which can generate that, okay? So that's the answer, okay? Thanks for that. Any other questions?
Okay, other questions?
So that's the answer, okay? Thanks for that.
Okay.
Fine. It's okay, I can hear you.
So for those species of elaphorans that swim to breathe, do you think there's a different type of system? Are they always synchronous in terms of the cardiac and the respiratory? Or is it the same system across all of them, or maybe depending on the strategy you have different types of systems?
Well, we haven't done a comparative study, of course. We just have the model animal. But if an animal, there's a guy called Satchel, Jeff Satchel, many years ago that I knew. He died many years ago. He was earlier generation than me even, so that would make him 120 years old now. He was interested in exactly what you've just asked, and he thought he could see the fact that when sharks are swimming, not flat out chasing, but just swimming, there's a rhythm that spreads from the front of the shark all the way back. And this looks like what we see because the ventilation muscles fire one at a time. So the jaws move, the operculum moves, not the operculum, the spiracle moves. Okay, sorry, and the gill arches. I work on Telios as well, and and then when the animal swims, it's swimming at a respiration rhythm. It swims as it breathes. That would work as well. Okay, so if there are slight accelerations and decelerations, if they relate to their opening and join. Now, that's never been proven, but I would guess that it could be. If sharks are swimming flat out, then we just discovered that they ram ventilate, okay? I'm never too sure about whether there's sharks that have to swim to breathe all the time. We did some work on sturgeon and discovered they're much more resistant to hypoxia when they're swimming than when they're not swimming. When they're not swimming, their operculum, they do have, doesn't close completely. So when they're swimming, they're getting better ventilation, okay? They're more effective ventilation of the gills, and if you make them hypoxic, they maintain oxygen consumption to higher levels at hypoxia, you understand? So there are things going on between swimming and ventilation which are interesting, and they're closely related animals, of course, more or less. So there are things going on between swimming and ventilation.
No more questions? Okay, another question?
Hello.
Hello.
Just wondering, has any of your work in physiology been linked with the stress after capture? Do you know if there's any implications on the cardiorespiratory system that you've studied when you catch and release a shark or something like that? Do you know if there's any
catch and release sharks?
Yes, so stress after capture.
Isn't what we do, but there's certainly an enormous amount of. We have worked on stress and recovery rates in reptiles and what have you. Animals are traumatized by what we captured, anaesthetized. The point is the preparations I've been showing you weren't recovering. I didn't talk about the stuff that Holly asked about. We did that then. We had to leave the animals for days to recover and come back, creep back into the lab. Animals know if you're there. No, they do. So animals are stressed all the time by the fact that there are these monkeys working on them. So and the stress of being handled, particularly the way that you saw in that wonderful film of animals being taken out and measured, they will be traumatized. But you notice that the people work on them very carefully, gently put them back in the seawater, they swim away. They will take maybe several hours to recover from that stress, but they will recover under those circumstances. I had a student working with me who was very good with fish. She was a fish enthusiast, and we were physiologists. She would catch the fish from water in a plastic bag, remove it into a bucket and then bring it to the lab, so it never left water. The standard fish, oiks the fish out, throws it in a bucket. The animal is massively traumatized by that, and it takes hours to recover. And there was some very interesting work done by a Scandinavian guy called Soivio many years ago, traumatized by that. Fish physiologist sets an animal up at night, goes for a beer, goes to bed, gets up next morning, works on the fish. Now, I've seen some fish in pretty tragic situations in labs all over the world, because physiologists, but anyway, Soivio worked out that it took at least 51 hours for an animal to recover from anesthesia and insertion of a cannula. So if you're cannulating a fish, you should leave it for 2 or 3 days to recover. That's difficult because the animal may deteriorate. Physiology is tricky because if you're looking at integration, you want to get the real story. You don't want to be working. So your question is, in other words, is very interesting because if the animal's traumatized, you're not getting normal physiological. But what we're doing, the mechanistic approach I adopt with decerebrated, paralyzed animals is the kind of things that biomedical labs have been doing for years. I remember at a conference, a guy said to me, vagus isn't that important because vagal tone is very low in cats. And I said, no, it's only very low in anaesthetized cats. In a cat, vagal tone will be variable. and sometimes high. And so, you know, if you're a dedicated physiologist, you can lose contact with the animals in their native state. And I understand that this society is mostly about conservation and ecology, and I've got huge respect for that. I'm a lover of sharks. I was a scuba diver many years ago, and they're the most beautiful animals underwater. And as they rightly say, they're not dangerous. They're just wonderful. Anyway, thank you very much. I think that's
we all
probably
agree.
it. Thank you.
Again, thank you. Okay, we can move to the next speaker, Kolobe Monwa, with a presentation on gut microbial community structure of emerald sharks from the east coast of South Africa. Please. Presentation on gut microbial community structure of emerald sharks from the east coast of South Africa.
Okay, thanks, for the intimidating opportunity to open series of talks on biology of elasma branch. The talk is on gut microbial community structure of hammerhead from the east coast of South Africa. So I think we'll need to draw a knowledge from the bony fishes because there's little knowledge known on the gut microbiota of sharks. There's been a very good studies on the gut microbiota of bony fishes. They've been well studies using both cultured and wild populations. And from those studies, we know that they've been studied using both cultured and wild populations. We know differences in gut microbiota between cultured and wild populations in bony fishes. And we know that mostly the cultured populations are associated with what we call transient microbes. These transient microbes, they only colonize and proliferate under controlled diet and water quality. While in the wild populations of bony fishes, we've got what we call permanent gut microbiota, which are associated with the stable conditions where the fish is associated with. Basically, this is the stable environmental conditions like your salinity and sea temperature. So there's a known functional rules within the gut microbiota of the bony fishes, that these microbes, they play different biological roles, including the nutrients absorption and then development of the vertebrae in the juveniles. And one of the obvious reasons for this lot of research on the bony fishes is that these fishes, they've got high economic value in fisheries, so they've been well studied and researched. But we also know that there's been a long history of commercial shark fishery globally. So equally, the sharks, they do play a very important role in fisheries, but there's little knowledge on the gut microbiota of sharks. Our knowledge is only limited to one study where they only use 3 species of sharks. And here is just a background of the studies that have been done on the east coast of South Africa based on the scalloped and smooth hammerhead. These 2 studies, they use the stomach contents analysis and the tag recapture data. And they both found that the juveniles of the scalloped and hammerheads, they feed more at the coastal nasal areas on the small invertebrates, while the adults, they move or they swim further offshore for feeding. So based on this study, there's clear evidence of what we call ontogenetic partitioning of resources or diet within the scalloped hammerhead. We know that the juveniles, they feed at the necessary areas, and the adults, they move further offshore for feeding. So based on that knowledge, we have also seen that from bony fishes, the same fishes also, they undergo what we call ontogenetic diet shift. There's also found that the gut microbes between the juveniles and adults of fishes, they are different, because the fishes, they also the fish, they also partition diet and they habitat. So we want to, based on this study, my aim was to use the same knowledge to characterize and determine differences in the gut microbiota of the hammerhead sharks at a very fine scale, because we know there could be differences between species, but this was at a very fine scale at the congeneric level, firstly between the two species of hammerhead, scalloped and smooth hammerhead, and the second one was within species at the ontogenic level between juveniles and adults, given our knowledge that they partitioned resources, habitat and diet. And lastly was to identify the predominant or the core gut microbiota in both species. And yeah, for those of you not familiar with the African coastline, this is the map of Africa with the South Africa there, flanked by both the Indian and Atlantic Ocean. It's got 4 coastal provinces. The study was focused on the KwaZulu-Natal province, all the way from Richards Bay to Port Edward. So all these sites are subjected to the warm temperate waters of the Indian Ocean. So they're basically very stable permanent microbes, based on what I've told you, if they are subjected to the same sea temperature and salinity. That's why the study focus was at a very fine scale within species. And we used what we call opportunic sampling. So basically our organization, the KwaZulu-Natal Shark Sport, they use what we call shark control fishery to protect bathers along the KZN coastline. We sample this through what we call meshing. So meshing, we've got our crews that they launch the boats in the early hours of sunrise every weekdays. And as you can see there, they've launched their boat to check this shark control fishing gears. So, and they will use the shark nets and the drum lines. So all the animals that I've analyzed, they come from these fishing gears where we control sharks for bather protection purpose. And this is the 2 species I've told you about, the scalloped and smooth hammerhead. And the samples were basically collected from the proximal intestine to the spiral valve. So that's the stomach. So basically whatever we found in the stomachs, we discarded the prey items or anything. And then we squeeze the liquid between the proximal intestine and the spiral valve into the vial, and they were kept frozen within 3 hours of collection. And overall, this is our sample numbers. We collected 25 samples, 13 of scallop and 12 of smooth hammerhead. You can see there the ratio of juveniles and adults there. Overall, we had 13 juveniles and 12 adults between the 2 species. And DNA was extracted using mobile pulsar DNA isolation extraction kit. And of course, we sequenced the 16S ribosomal RNA gene tech sequence using the PET and sequence on Illumina MySequence platform to identify the operational taxonomic units. And sequences were quality trimmed and analyzed using the software quantitative insights into microbial ecology. And we did a normal test on the alpha and beta diversity in Chai. And also we ran the Shannon diversity index to determine significant differences between juveniles and adults and also between the 2 species. And here are the quick view of the results. That's the relative abundance of the microbes we found. So you can see this is between the 2 species, scalloped and smooth hammerhead. Protobacteria were predominant in both species, followed by the Firmicutes, Bacteroides, and Lactobacteria. And then between juveniles and adults, it was the same pattern there, where you can see that Protobacteria were very predominant, followed by Firmicutes, Bacteroides, and Lactobacteria. And then on the statistical analysis, we found that there were basically 389 OTUs, operational taxonomic units between the 2 species, mostly from the smooth hammerhead, but we find the same OTUs in the scallop hammerhead. And also the analysis of similarity revealed no significant differences between the microbes, between the life stages, adults and juveniles of both species, and also between adults and juveniles within the same species. And what was interesting was the core gut microbes. As you've seen, the predominant microbes were Protobacteria. But within the Protobacteria, the Vibronales, the order Vibronales was most dominant. It was found in abundance across all 2 species and across across the 2 life stages, adults and juveniles. And they were followed by the 2 orders, the Pseudomonales and the Aeromonadales. They were also found in abundance. But what is interesting is that we had the Lactobacillus, which were only found in the smooth hammerhead. They were also found in abundance. So in conclusion, we could say that basically there is no significant difference in gut microbiota at the ontogenetic level between adults and juveniles. We could say that and also between the two species at the interspecific level. But what it is interesting is that the protobacteria are the dominant gut microbiota in both species and even at the life stages. And the same pattern has been found in the bony fishes where protobacteria were predominant. And also, what is interesting is the relative abundance of protobacteria. What is interesting is that you find different protobacteria at different level of abundance. And in this case, we found that the Vibrio and Pseudomonales were the core protobacteria between those two species and even within at the life stages. And then the Lactobacillus were the core formicort species. So based on this study, we can see that although the protobacteria are the core gut microbes in these sharks, but the level of abundance is very important. So future studies need to focus on the significance of the biological role of the abundance of the different orders of the protobacteria. Thank you. Future studies need to focus on the significance of the
Okay, thank you, Caleb. Questions? Okay. Yeah. Okay, thank you, Caleb. Questions?
Hi. So at the beginning of your talk, you said that different microbes will have different functional roles. Do you know if the difference you found between that there's any different role for those bacteria have different guts of the shark? Do you know if the difference
Come again?
Sorry. So the different, you found different microbes in both sharks, right? Does that tell you any difference in the function of the microbes in the gut of the different sharks? Right? Does that tell you any difference in the function of the microbes in the gut?
At the functional roles of those microbes, yeah. No, we didn't look whether we know that the protobacteria are mostly important in the breaking down of complex biomolecules. So that's what we know generally, that the protobacteria, they play a role of breaking complex biomolecules. So, but to what extent they break the prey items within sharks, yeah, we have not checked that. So it could be next focus. So, but to what extent they break the prey items within
Okay, no
sharks,
more questions. I have a curiosity. Okay, no, here there is another question. Sorry. Here.
Are the adults fish feeders? And the adults that go offshore, they're feeding on fish?
Yes.
Oh, okay. So I guess they'd pick up the gut bacteria from their prey anyway, wouldn't they? But anyway, yeah, thanks.
Is that a question or comment?
Take also in consideration on the differences in the water where you capture the fish, the composition of the water, if there are...
The weather.
Yes.
The water where you...
Or the water.
Yes.
The water.
Water or weather?
Water.
Water. So you mean the water temperature, water quality, or water or weather? Water. So
you
mean
show
the
that
water temperature, water quality, or
the fish in a different place. Yes.
yeah.
So you take also into consideration the quality of the water in terms of Pseudomonas or other parasites that you find.
No, we didn't do that. Yeah, because as I told you, that this was the aim here was just to pick any differences in gut microbes at a very fine scale. That's why it was within species between juveniles and adults, or in the congeneric. I mean, these are two species from the same genus. So to expect difference, the feeding behavior, it might be the same. So this was a very scale. So and here can you see all the size, they are subjected to the same water temperature. Basically, the sea surface temperature remains constant between all the size. That's a stretch of 320 kilometer coastline. Basically, the sea surface temperature remains
Okay, Okay, thank you again, Colop. And then we can move to the next
Okay, sure.
presentations. Right, thank you. Okay, our next speaker is Licia Finotto. What's my destiny? Reproductive impairment following fishing capture stress in an Australian constrictian, the southern fiddler ray Trigonorrhina dumerili. Please. Fishing capture stress in an Australian constrictian, the southern fiddler ray Trigonorrhina dumerili.
So, good afternoon and thank you everybody. Today I will present you some of the results of my PhD, which is partially aimed at investigating the reproductive consequences of fishery stress. So we all know that, unfortunately, the vast majority of sharks, rays and chimeras are in a bad conservation status, and that fishery represents the main threat for these species. Not only because they are subtracting animals from the population, but also in terms of bycatch, which is captured, undergoes the stress, but then is released back in the water. The problem is what happened to this animal. For a long time, this was completely a mystery. Thanks to dedicated research, now we know that in some cases there is even a large amount of sharks that doesn't survive, and that's an important information that needs to be taken into consideration while developing the management measure for this population. But even in the case a shark is lucky enough to survive, the injuries and the stress they undergo during the fishing operation and the consequent physiological, behavioral and energetic alteration can determine severe long-term consequences that in teleost has been proven to impact growth, immunocompetence and reproduction. On chondrichthyon, data are basically non-existent, and that's why I was mainly focused on reproduction, given that it's the process through which population can contribute to the next generation and maintain their abundance. To do so, we decided to work with southern fiddler ray, which is an endemic viviparous species of southern Australia, and it's a common bycatch. That means that if any reproductive consequences actually exist, there's the possibility that a large number of animals might be impacted by them. But also, despite this species is not at risk of extinction, is closely related to the large guitarfish and wedgefish that have been recently listed as like the most endangered fish in the ocean. And any useful data that can be gained from studying these species might help to improve the management of also of these other species that are more difficult to study. To do my experiment, I collected pregnant female from the wild. I transferred them in a housing facility where they were maintained up until parturition. And all the females were divided in two groups, the trolled group and the control, obviously. And the troll group underwent trolling simulation for 7 hours and air exposure for 30 minutes as to mimic the condition during a commercial operation. And to simulate trolling, I placed the animal in a net, in a huge tank. And thanks to the movement of a wood panel, we were able to generate a current that was simulating the condition in which the fish is when it's trolled at sea. After 7 hours, we simply placed the animal in an empty tub for the air exposure part of the experiment. I then focused my interest mainly on neonates and neonates characteristic. First of all, I recorded the time in which they were born, which all happened between April and May. This is also the in literature, and therefore this seems to suggest that fishery simulation doesn't induce parturition as it does for other more sensitive species. I was also interested in investigating whether there was a different occurrence of stillborn neonates between the trolled and the control female, but I didn't find any difference. So even in this case, the fishery simulation doesn't seem to affect this trait. At birth, I collected a blood sample from all the neonates, and I used it to produce some blood smear to calculate the granulocyte to lymphocyte ratio. This is an important proxy that has been proven to be related to stress in constriction. And I found that trolled neonates born from trolled and therefore stressed mother present a significantly higher value for this ratio, which means that they are already stressed and they are mounting an immune response to try to deal with this stress. Obviously, I also collected some of the most important parameters, body mass and total length at birth. And in this case, I again found that for body mass, troll neonates are significantly lighter. And thanks to using an ultrasound, I was also able to evaluate the volume of the residual yolk sac. And even in this case, though, despite there seems to be some difference, there's no significant difference between the troll and the control neonate, and the large difference that can be seen there is mainly driven by difference in body mass, because obviously larger neonates have also a larger amount of yolk. And then I tried to evaluate a growth rate. To do so, I measured again body mass, total length, and the volume of the yolk sac every 10 days from birth to 1 month after their birth. And as expected, body mass and total length was increasing while yolk sac decreased. But when I compare the rate with which these different parameters were changing, a significant difference was found only in total length, with troll neonates being smaller and shorter after a month after birth, indicating a possible reduction in their growth rate. Then I also tried to evaluate some neonates' performance, among which their swimming ability. To do so, I placed the animal in a corridor and I elicited their swimming behavior by tipping their tail. I recorded their performance, and then I measured the mean speed. And given to the fact that basically no neonates swam from the start to the end in a single movement, but tended to stop at different length from the start, I also recorded the length of the first segment that they covered. Control and troll neonates doesn't differ for their mean speed, but the troll neonates swam for significantly shorter length. So in conclusion, what we found is that troll neonates have both a smaller body mass and a smaller at birth and a lower growth rate, and this is happening despite they use basically the same amount of yolk as the control. And this is probably explained by the fact that the GL ratio is higher, therefore they are stressed and they're using part of the energy not for growth, but to deal with the stress and the immune response. Therefore, like being smaller can be a quite deleterious consequences, because we know that in the ocean, big fish eat small fish, so being smaller might increase their chances of being predated. We also found an impaired swimming ability. Again, this can affect their predator escape ability, because if they swim for a shorter distance from the predator, they might not be able to hide, or they might not be able to outrun the predator itself. But in this case, an impaired swimming might also affect their ability in catching the prey. So in conclusion, all this data seems to suggest that neonates born from mothers that have been stressed and fished during their pregnancy can other that have been stressfully affected survival, which in turn can affect parental fitness, but also on a larger scale the population and the population abundance. Obviously, all these should be tested in a natural environment to see if survival is actually affected, and other species need to be investigated. I would like to thank all my collaborators and also the EEA board and the Save Our Sea Foundation for the student bursary that they gave me. Thank you.
Okay, thank you, Licia. Thank you. Questions? No questions? Prof. Taylor, a question?
Very nice experiments, I think, and I think very worthwhile.
Questions?
The other thing that fish get when they're trawled, of course, is being crushed, isn't it? Because there's and of course you're not crushing them. So I should think the trauma of being trawled would be worse than what. But I think your study was well worthwhile.
Yeah, in my case, I placed all the animals together to simulate the crushing. Because I was also investigating their metabolic rate, and I had just one respirometry chamber, so I had to be forced to stress them one each in the net. But to try to compensate for the absence of crushing, I prolonged the trawling simulation for a longer time, because usually commercial trawling are like trawling the animal for around 3 to 4 hours, so I extended the period of the fish in the net to try to compensate for the fact that it wasn't like the pressure of other animal in the net.
Okay, other questions? No? I have just a curiosity. Have you some indication on the putative morphofunctional alteration in tissues and
in what sense? Sorry.
if there are evidence of functional alteration, the putative effect on morphofunctional.
Not for sharks. I mean, there has been another student working with the same species in my lab previous to my experiment, but for sharks, there's no information at all. The only information on their reproductive impairment is related to induced parturition, which is quite common for some species, but there's nothing on neonate survival or neonate performances. There's much more on teleos. That's why we should improve our knowledge in this.
Okay, thank you.
Thank you.
Thank you again. And okay.
Thank you.
Our next speaker is Andrea Bellodi. Growth and edge validation of the Thornback ray in the West Central Mediterranean Basin.
Okay.
Growth and edge validation of the Thornback ray in the West Central Mediterranean Basin.
Sorry, thank you. Good afternoon. The present study deals with the growth and age of the Thornback ray, and has been carried out through the collaboration of the University of Bari and also the COISPA Institute. So, the Thornback ray, is a medium-sized skate widely distributed in the Mediterranean and all the eastern coast of Atlantic. And among other elasmobranchs of this area is one exploited, as frequently landed as bycatch. So this situation, among others, led the IUCN to classify the species as near threatened. For this reason, of course, the need of proper management plans seems even more urgent. And however, this kind of plans, as you know, requires a lot of data. Particularly important are those regarding age and growth, indeed. However, despite the widespread distribution of the species, this kind of data are still lacking for the thornback ray, especially for the Mediterranean population that seems to be far less studied than those inhabiting Atlantic coast. Indeed, only 3 studies are present up to date on this aspect of the thornback ray life history. Moreover, no direct validation attempt where the validation studies are being attempted yet in the basin. With this aim, we choose to sample a very wide area in the central Mediterranean Sea that involved 4 different FAO geographic subareas, dealing with more than 700 skates, well distributed in sexes and sizes. From each individual were extracted the vertebral centra that were treated in order to obtain the best suitable possible section for the age reading. The readings were carried out following the newest handbook provided by the GFCM, and of course were counted only the translucent bands, starting from the very first after the beer mark. In general, the band visibility was very high, and thus allowed us to state that no staining methods were required or should be employed for this species, and also allowed us to reach very good values in terms of reading precision and reproducibility. Moreover, from each vertebral centra was also recorded the age type in order to perform the marginal analysis. As you can see from the chart, a very good deposition pattern was found, with the opaque band which is laid down mostly during the warm month, while the translucent during the coldest, of course. So this validated the growth band deposition periodicity for this species. Moreover, considering also that not always the common von Bertalanffy growth function has been proven to be the most suitable, the most precise to describe elasmobranchs growth, and this is mostly true for bathoids. We choose also to apply to our age length data also 4 different growth models. 2 of them that deals with simple curves, like the 3 and 2 parameters von Bertalanffy, and 2 of them that deals with an S-shaped curves, the Gompers and the logistic ones. However, as you can see from the tables, in this case, both for females and males, the common von Bertalanffy function have been proven to be more precise in terms of fitting to the age data. For this reason, the next step was naturally to apply this kind of function to the to all observed data, divided, both for sexes and combined sexes, and also divided by each different subareas investigated. It's interesting, and here you can see all the computed results. It's interesting to note out that the Chen test did not show any statistical difference between the growth in all sexes, in all the investigated GSA, which is kind of strange. And however, statistical difference were found between growth of the species from the different GSA. As you can see, with skates from GSA 19 and 11, that seems to grow a little slower. In addition, for the first time in the Mediterranean Sea, was also carried out a tag and recapture experiment. Skates were tagged both with a external tag, the spaghetti ones, and a chemical tag, an oxytetracycline injection. A total of 98 specimens were tagged and successfully released in the at sea, and 2 of them were captured after almost a year for both of them of freedom, of course. From the analysis of the vertebral section, of these 2 recaptured specimens, we found a very visible, well-visible oxytetracycline mark. As you can, it's a bit dark, I think, and followed by the subsequent deposition of the complete annulus. As you can see, a very wide opaque ring there, and I don't know if you can. There is the beginning of the deposition of the translucent one after. Thus, considering this, that a whole year was passed prior to the release, allowed us to validate the aging criteria utilized in this study. In conclusion, the Thornback ray skate is commonly reported, of course, in literature for other elasmobranch species, appear to be a slow-growing and long-living species. However, differently from the majority of this animal, no difference in grow rate were found between sexes in all the investigated GSA. Moreover, however, the difference in growth that were found actually in the growth rate of the skates that comes from different GSA could be related from the different the different environmental conditions that characterize in this area, of course, and of course further analysis should be welcome in order to solve this aspect. While the high visibility of the oxytetracycline mark found in the 2 recaptured individual testified in some ways the good suitability of the employed dosage for chemical tagging. And in conclusion, our results could represent a very good step forward, a better comprehension of this important bait catch species, investigating a very wide area in the central Mediterranean Sea, and providing new age data, very solid, validated also both by semi-direct analysis like the marginal analysis, and for the first time again in the Mediterranean Sea, also by direct methods as the chemical tagging. Thank you. Sorry, I'm a bit in rush.
Okay, thank you, Andrea. Thank you very much. And a question? Okay, thank you, Andrea. Thank you very much. Okay, here there is a question.
Has this kind of analysis been done on species in other regions? And
Sorry.
Sorry. Has this kind of analysis been done on species in other regions? Has this kind of analysis been done on the species in other regions? And if so, how do your results compare with what's been seen out?
Yeah, we followed our protocol and we.
If so, how
yeah.
do your results? Is this similar growth rates?
Not with skates in Mediterranean.
Yeah, we followed our protocol. I'm sorry.
Not with other skates in Mediterranean.
With the same species in other areas outside the Mediterranean.
Not that I remember. Not with other skates in Mediterranean.
With the same species
Okay,
in other areas.
another question.
Not that I.
Thank you very much for your presentation. Did you use scanning technique or?
Yeah, we tried, for example, the red alizarin and so on, but we didn't find any improvement to the band visibility that were generally band well visible without staining. So we choose to save time and equipment.
Okay, thank you.
Without staining, so we choose to save time.
Okay, okay, no more questions. Thank you. Thank you again, Andrea. And we have the next. The next speaker is Tal Starostineski Malonik, blood biochemistry and hematology of dusky sharks Carcharinus obscurus and the sandbar sharks Carcharinus plumbius in the eastern Mediterranean Sea. Please. Carcharinus obscurus and the sandbar sharks Carcharinus plumbius in the eastern Mediterranean Sea. Please.
Okay, hello, I am Tal. I'm from the Maurice Kahn Marine Research Station that belongs to the University of Haifa, and I will talk, as said, about the serum chemistry and hematology of the dusky and sandbar sharks in the eastern Mediterranean, meaning the coastal waters of Israel. So why test blood? The serum chemistry and hematology profiles are a very strong tool to assess the physiological status and the health of individual and populations of animals. So establishing the reference intervals for these analytes of individual and populations keystone species and ones that we want to conserve is also important to improve their conservation and management. Why these two species of sharks? Well, we all know sharks are top predators, and they're very important for the health of the ecosystems they live in. And yet, no reference intervals have been established for many species that live in the Mediterranean Sea, including these two species. The dusky sharks are defined by the IUCN Red List as data deficient in the Mediterranean, and the sandbar sharks are defined as endangered. In the coastal waters of Israel, we have a very special phenomenon where sharks aggregate around the outflows of power plants, coastal power plants, where there is hot and mostly saline water coming out into the sea. One of these power plants is the Hadera power plant, which is the study site. And to use this very good opportunity to study the animals that aggregate there, the study site station has been conducting catch and release tagging and sampling activity. In this site, every winter between November and May, there are a few dozens of sharks that aggregate in this spot. They are mostly predominantly dusky females and sandbar males. This last winter was the first time we caught and tagged dusky male, but other than that, it has been mainly dusky females and sandbar males. The duskies are a bit bigger. They are between 2.5 and 3.5 meters. The sandbar males are smaller. They are around 1.8 meters. And to hear more about this phenomena and about the activity of the research station, I highly recommend that you attend the talks of Aviad Shainin, Adi Balash, and Ayad Beagle tomorrow at these stated hours. So what is being done is the sharks are caught using lone line or drum line, pulled to the side of the boat, strapped and tied and alongside the boat, flipped on their back, so blood is drawn from the caudal ventral side of the shark, like you can see in the picture, using a syringe and a needle. The blood is placed in tubes which are kept cold until we reach the coast. Then blood smears are prepared and stained for differential white blood cell count. The serum chemistry is analyzed using an automatic analyzer, and the hematocrit is tested through centrifugation of heparinized capillaries. To this point, so since 2016 and until today, and also next winter, this upcoming winter, we will also be collecting samples, so our data pool is expected to grow. For serum chemistry, we have analyzed 15 analytes for 19 dusky sharks, from which, apart from one, were all females, and 12 sandbar sharks, which were all males. The blood smears and the hematocrit test we've only started performing this last winter, so the sample size is still quite small, and that is why I will not speak of the hematocrit results yet. So the serum chemistry results we have so far are thus. You can see that the urea, chloride, and sodium values are quite high, as expected for sharks, and their distinct physiology, which they use urea for osmoregulation purposes. The analytes marked with an asterisk are the ones where there was a significant difference between the two species. Also, it's important to note that we removed highly hemolyzed samples from the data because it affects the values that we get in the results. I will further discuss a few of these results. So for both of the species, 11 out of the 15 analytes were never described before. The GGT activity has never been reported in sharks, not just in these two species. Glucose, cholesterol, and CK levels had a significant difference between the two species. The CK activity was higher in the sandbars than in the duskies, but both species levels were higher by 1 or 2 orders of magnitude than what was reported for other shark species. And this could be possibly due to the muscle damage that was caused during capture. The other studies that tested for CK used either a lasso capture with a rope or a trolling net, so there was no muscle damage as it was with us. Also, AST activities of both the species were significantly higher when compared with Atlantic sharpnose, spiny dogfish, and sand tiger sharks. This is also possibly related to the muscle damage caused during capture. The total protein levels we've measured for both species were significantly higher than those of sand tiger, bonnethead, and whale sharks, which in studies that use the same measuring method, the Burette method. There were also other studies that used other methods, but these affect the results, so we couldn't really compare the two. Also, protein measurement is affected by is impaired by the high levels of urea. So also there needs to be further thinking about the right way to measure that. Amylase activity was measured by one other article in other sharks, but again, they used a different measurement methods, which affects the results, so it wasn't comparable to ours. For the hematology result, these are pictures from blood smears of the one male dusky that we caught this last winter. You can see the different leukocyte and thrombocyte cells, and this particular individual had quite a lot of heterophils. And this is the summary of the counts that we've performed so far, the differential count of leukocytes and differential count of the thrombocytes. Also here there is a difference between how different studies describe or count their cells, because sometimes they count basophils that we didn't, sometimes they don't count the young granulocytes that we did, sometimes they include the granulated thrombocytes with the other leukocytes because their function is still not known. So again, there is kind of a difficulty to compare with the results of other articles here. So to summarize, we have described a few analytes that were not described before. For the most analytes, the levels that we measured were in accordance with what was previously described, or with the known physiological traits of these sharks. There were several differences between the two sharks that we examined. There was a possible influence of the muscle tissue damage that was caused during the capture on several analytes, CK and AST, as we've seen. And we have encountered several challenges when trying to compare with the results of other studies, because there is a variation in the biochemical values that come from differences in species, in geographic areas, in ecological habitats, in genetics. Different sexes, different life stages show different values, and because these are such sparse animals, every study takes advantage of the local population that it has access to, and it's very hard to know where these differences are coming from. There is also different measurement methods between the different studies, which affect the results and the ability to compare. Also, we've seen it with amylase and total protein. And there is a lack of an accepted standard for the descriptive statistics that we use to define these intervals. Some researchers use median versus mean, some studies use standard deviation or standard error, some use the quartiles and the ranges, and again, it's hard to compare. Thank you.
Okay, thank you, Tal. Thank you very much. There is the time for a discussion. Okay, thank you, Tal. Questions? Thank you very much. There is the time for I have a question. Did you use commercial kit for your analysis?
We used an automated analyzer for the, you
But
mean for the chemistry, for
yes,
the serum chemistry? An
commercial.
automated analyzer. So not a kit, but an entire machine.
Okay, not commercial kit for fish are available, I think. No?
It's not. No, it's not. It's a veterinary laboratory that does this for us, and they use an Roche automated analyzer.
Okay, okay, thank you.
Sure.
Okay, there is another question. Okay.
What type of coloration did you use
there
for
is
the
another
blood
question.
smears? What was the methodology?
So actually, we do like a double blood smears. I do them at the station, and also, we send whole blood to the veterinary lab, which also perform. So we both use Romanovsky stain, but they use a different solution, an automated stain, and I do it diff quick manually.
Okay.
But yeah.
Perfect. Thank you.
Theirs is better.
Okay. Another question? No?
Theirs is better.
Okay. Another question?
If possible, I have a question for you.
Oh, sure.
Yeah. You mentioned in your conclusion that there is the possibility of influences from sex differences on the data you collected. But you divided between males and females. I saw you somewhere in your slides that you divided in males and female. Did you find any correlation with sex about your data? You could find correlation between different population of your data and female. Did you find any correlation? Or both males
again,
and females are overlapping in their data?
Yeah, because as I said, there are many factors that can affect the results, and the fact that we have males from one species and females from another species also makes it hard to tell which one is the one influencing here. And also, again, their size is different, their sex is different, the species is different. So even though they live in the same, you can't really tell which one is the one making the difference. And also with other studies, it's even more because they don't differentiate the sexes. Sometimes it's juveniles, ours are adults. There are many factors that can affect that, and so it is hard to compare. Thank you.
Thank you. Okay, the next talk is by David Ruiz Garcia. Paired laser photogrammetry is a non-invasive technique for estimating shark body size and weight. Paired laser photogrammetry is a non-invasive technique for estimating shark body size and weight.
Hello everyone.
I'm David, and I'm going to present the outcomes of my research on the use of period laser photogrammetry as a non-invasive technique to estimate shark body size and weight. So first of all, why is it important to know the size and the weight of a shark? So in natural populations, it allows us to calculate the growth rate, assess changes in body condition, determine the maturity stage, and differences among populations, which is also important to know the dynamics of these populations, so that we can establish effective management strategies. But it's also important to know it in aquariums, because we can detect unexpected changes in their body condition as well, monitor seasonal fluctuations related with sexual development or ontogeny, and also determine food requirements, medication doses, and anesthetic doses. Okay, but how can we get this information? Traditionally, it was done through invasive methods which require the capture and handling of the animals. However, this produced gowns, deformities, even premature birth and abortion. So we need, well, this hampers conservation efforts, obviously, and we need to create remote measuring techniques. Period laser photogrammetry has been suggested as a cost-effective and non-invasive technique to obtain morphometrics from sharks. Basically, you have two lasers which are parallel, so you always know the distance between the 2 points of light that they project. You project those points of light upon the body of the animal, and you take a picture, so you have a scale bar within the body of the animal, and with a simple rule of 3, you can calculate any morphometric. But how can you get all the morphometrics that you want, and how can you get weight from this technique? Okay, so these length-length relationships and length-weight relationships relate the growth rate of a morphometric that you can measure and the one or the weight that you cannot measure directly. It has been largely used in fisheries, and it has proved to provide good estimations for either weight and other morphometrics. However, they usually use the precaudal length and fork length to estimate total length, and total length to estimate the weight of the sharks. However, these may not be the best predictors, and also, as we will see later, it's difficult to obtain these morphometrics from using period laser photogrammetry. So we consider a larger selection of variables. Our objectives were to test the accuracy and precision of period laser photogrammetry, determine the effort requirements for the proper implementation of this technique, and to provide a resource for the estimation of shark total length and body mass. Okay, so how did we do this? We collaborated with 3 aquariums, the Oceanographic of Valencia, the Zoo Aquarium de Madrid, and the Aquarium de Sevilla. And we used 5 species: sandbar shark, 18 specimens, sand tiger shark 8, nurse shark 6, nurse hound shark 10, and short-tailed nurse shark 7. And the equipment, it was 2 underwater lasers, which were mounted onto a copolyester custom design platform to which we drill Allen screws to adjust the lasers in parallel in the underwater camera. And then we tested the accuracy and precision of the technique, so that we calculated the magnitude of the different errors that can occur. A common error is the non-parallel alignment of the lasers. Since the lasers are not parallel, the distance between the 2 points of light will depend on the distance to the target upon which you are projecting the lasers. So we ensured that this distance was always the same by putting an object, a length-known object, at 2, 5, and 10 meters, which is the distances to which we are working with the sharks. And we ensure that the distance is always the same. And parallax error is not completely perpendicular to the laser setup. So as the degree of deviation increases, the perspective of the lasers also increases. So you have an error in the scaling process. But we observed, measuring different objects of length-known size, that when the deviation is lower than 10 degrees, the error is lower than 1%. So we can still use the images in which there is some deviation, but when it is larger, we have to discard that picture. And the last error is the operational error, which is the simple error that you commit when you are measuring in the software that you use. But we observed that this error is minimal by measuring different objects of different sizes at different distances, So we can conclude that this technique is highly precise and accurate, and we complete the first objective. Now we can take the measurements of the shark, but what measurements do we want to take? So understanding that volume is a good estimator of weight, we thought, okay, let's divide the body of the animal in different geometric figures. And how can we calculate the volume of these figures? So we will use these morphometrics. So we came with a set of 18 morphometrics for each species that we measured. And the divers went into the tank, and they dive on the surface. When they saw the shark that they were targeting, they dive down, and they position themselves in perpendicular position to the shark, projecting the lasers upon the flattest area of their body to avoid parallax error. Once we have the footage, we identify the shark that it's in the footage, and we extract the photograms in which the parallax error is minimum, and we take the measurements using image J. And regarding the obtention of weight, we couldn't do it with ram ventilator sharks because it may threaten their health. So we didn't do it with sambar shark and sand tiger shark because it's a pilot project, but we aim to do it in the future. Basically, for the other species, we catch them with a hand net or a stretcher, and we measure the weight in a scale. And what did we get? Okay, so for the second objective, which was the determination and we measured the weight and implementation of the technique, we calculated the success rate as the number of successful encounters divided by the total number of encounters. We had more than 300 encounters during 7 hours of footage that we collected. And the success rate was a 78%. And the main cause of failure was parallax error. We also calculated this rate for each of the morphometrics. So basically, how many morphometrics can we get from an encounter with a shark? So total length, forelength, and precaudal length, which are the ones that are used in fisheries and are larger, the rate is very low, under 50%. So these measures are not very good for these kind of estimations. But the average was always about 60%, considering these low morphometrics as well. So it's good, and especially high for nurse-hound sharks. This is because nurse-hound sharks just light in the floor of the tank, exhibiting very calm behavior, so it was quite simple. For sambar sharks and sand tiger sharks, it was a bit complex because they need to swim constantly in order to breathe, so you have to catch them completely stretched. And for nurse shark, it was also a bit complex because they exhibited nervous behavior when the divers were in the tank. And for short-tailed nurse sharks, it was complex because they tend to accumulate all together, and yeah, it's a bit complex. Okay, so the relationships. For the estimation of total length, in the case of sambar shark, it was fork length, the best predictor. And we compare with the studies done in the wild, in fisheries, and the results were very similar, which indicates that we are doing something good. But since this is a long morphometric that we don't want to use, we calculated whether we could use a shorter one. And the height of the head was the best predictor, also providing very accurate estimations. For sand tiger shark, it was a combination of both the distance between the pectoral fin and the pelvic fin, and from the pelvic fin to the caudal fin. And for the others, it was the distance between the pectoral fin to the pelvic fin. And for all of them, the predictions were quite good, as you can see with the R square and the relative error. For the estimation of weight, in nurse sharks, it was the length of the head, which made sense because these animals have a very powerful suction musculature. And for nurse-hound shark and short-tailed nurse sharks, it was the head of the thorax, which is where all the organs are enclosed, and also the musculature of the caudal fin starts. Okay, so we have very accurate, precise estimates with low relative error and everything, but it's true that our variables were highly correlated, and we had a low sample size. So we are working in increasing the sample size and consider other factors as well. That's why we are creating a network of aquariums that want to participate in this project in order to obtain more information. So if any of you is interested or know someone that may be interested, you can contact with me, please. And okay, so we can conclude that period laser photogrammetry is a highly precise, non-invasive, and cost-effective technique. And the procedure that we used was also effective. And we obtained accurate total length estimates with short morphometrics, also accurate body weight estimates. And therefore, this is a useful tool in aquariums and potentially in natural environments. Thank you very much. And also thank you to the Erasmus Branch European Association Board and Safe Our Series for the student bursary.
Okay, thank you. Thank you, David. Okay, we can open the discussion. Okay, there is a question. Okay, thank you. Thank you, David. Okay, we can open the discussion.
Thank you very much for this presentation. I am in the relationship between your morphometric analysis and body weight prediction in relation to the what factor the liver size may play when you are going to extrapolate values for body weight, due to the fact that the liver, because of its high percentage of fat, of oil, is a major factor in the density of the animal and the weight. Thank you.
Well, these kind of relationships doesn't consider like specific organs. Is the this makes a growth rate of one morphometric in relation to other one or in relation to weight. So usually, as the shark grows, it grows proportionally according to weight and according to other morphometrics. The liver, it's a factor that also weight is a part of the body that affects, but we haven't considered whether it has a special effect or not in this relationship. And we wanted to consider as a factor the density of the animal, but it's quite complex to get this data since we aim not to touch the animals or anything because we were working in a public aquarium, so it's hard with this. So we couldn't use that factor.
Just a short comment, because it may be recalled some old, maybe very crude experiment, but if you take a shark is flotating, if you take away the liver, it's a very crude experiment, but it is very much related to the function of the oily liver in the story. So I would recommend to take into consideration in your further analysis. It should be not difficult, I think. Thank you.
Thank you.
Okay, thank you. We can move to another question. Just okay, thank you. We can move to another.
How did the shark react to the lasers in the aquarium? Because I thought this was fascinating. I think this is going to be so useful in the field. But do you worry that wild sharks that aren't used to divers and such would be scared away?
That's a good question. The first reaction, they don't care. But when they learn, they hate it. They run away. Yeah, it's very curious because they learn about it. And yeah, it's also because the diver is following them and they want to. They are like, leave me alone, you know. But yes, but this technique has also been used in the wild, and I don't remember if they reported whether the shark shows some behavior reacting to the lasers or not, but it's a point that may affect as well. Yeah. The shark shows some behavior reacting to the lasers or not,
just
but it's a
the
point
last
that may
question.
affect as well.
Yeah, I agree with what you said, that in the wild it hasn't been any information that it affects at all. We have used it with rays and it doesn't seem anything. But my question was, did you check any changes in any of your relationship between females or males or little ones and big ones?
We wanted to, but the thing is a small sample size, so it didn't make sense because, for example, we had for the nurse shark we had 2 females and 5 males. So you can find something, but it doesn't have to be a real result. We need to increase the sample size. So you can find something, but it doesn't.
Okay, thank you again.
Thank you.
The next speaker. Is Arconcia Katspirou, morphometric analysis of the Thornbeck ray raya clavate from the Eastern Ionian Sea. Please. Morphometric analysis of the Thornbeck ray raya clavate from the Eastern Ionian Sea. Please.
Hello everyone, and I'm here to present you a part of my thesis as well, which is carried out on skates and rays in the Greek Ionian Sea, starting with a morphometric analysis of the Thornbeck ray. As you might know, Raya clavata, a very widely distributed species, and is a very abundant species and dominant in commercial landings. Therefore, it is very well studied. But data on the morphometric characters are very scarce. Usually there are information on the comparison between total length and disc length, or total length and disc width. Why should we do the morphometrics for Rahida? First of all, to describe the interspecific variation in the morphology of the Thornbeck ray in the Eastern Ionian. Then first of all, attribute to the sexual dimorphism of the species, and to compare the characters between sexes, as well as to test the allometry of the characters measured. The samples were taken between 2017 and 2018 from experimental surveys, mostly MEDIT's program, but also from small-scale fishermen. And I could have a total of 140 specimens collected till today. The characters that I wanted to measure were about 35. I did some other measurements which are not included in here because they have to do mostly with the biology and reproductive biology of the species. And these are the analyses, the statistical analysis you can see. I will show you later. So this is the studied area where from north of the Ionian till south, with 77 females and 63 males. This is a scheme I made for my convenience in the lab, so I can see what I have to measure every time, because sometimes I forget, there are too many. As you can see, there are so many different characters we can use to do the morphometric analysis for the species. I combined many protocols from all over the world I found in literature, and I thought these were the most important for the species, for the Rahid species in the Eastern Ionian. So the most important descriptive statistics for the species are presented here in this table, which I could compare them with other areas. Mean total length showed no difference between males and females. Therefore, I used the equation between total length and disc length, showed no difference between length of those animals that they were missing part of the tail, of the whole tail, because of injuries or other reasons. And when I of the whole tail males and males total length to total weight, I didn't find any differences, while other areas did. And these are the morphological characters I plotted against total length and compared between females and males, and I found all these 13 characters being different between the sexes, such as the pelvic fins, which you could see with the naked eye. They were very different from males to females, possibly due to reproductive reasons, where the males need possibly larger pelvic fin because of their claspers. The p-values I found for females and males were positive allometric. They had high values, but they were higher in the Eastern Ionian. And I could show you all the allometries I did, but there was a limited time to show you the tables for the negative and positive allometry. But all I can say, it was very similar for females and males, with 9 characters being negative growing against total length, while 10 characters showed a negative allometry for males. And therefore, to conclude, that was the first attempt to describe the relative growth of several characteristics of the Rhakia clavata, where there is a serious gap of knowledge on the species. The maximum size of females and males in this study area was found a bit lower than other values from the Eastern Mediterranean Sea, but this can be attributed to the seasonality, the selectivity of the year, and the depth of the surveys. While the ANOVA showed us the differences in the mean values of, there was no difference in the mean values of total length, so we could use the total length of the whole population to find those missing lengths in the samples. There was a positive allometry for the length-weight relationship, but there was not statistically significant difference between the two sexes, which we didn't find in other papers surveys to show this, that we are in agreement. That's possibly because I was lacking large females and some large males, so I need to cover in 2019 these length classes, and the coefficient B had higher values for males than for females than males, and that's in accordance with other studies in the Mediterranean Sea. Most of the morphometric characters for the allometry now were strongly correlated with total length, but the nature of allometry differed between according to the character. The relationship of 13 characters against total length varied significantly, and this shows a significant variation in length in general, in growth in general. So in future studies, we would like to see the importance, this is of major importance, to increase our knowledge for the stock state in relation to environmental conditions. And morphometry has been proved to be used in separating species, populations, and races, and in determination of the sexual dimorphism. Therefore, I hope this data collected for this study can contribute to the stock assessment and conservation of the species in the future. And I would like to thank all my colleagues in the lab for all their help and support, which I could not do it by myself. It takes a lot of time to collect and measure all the species. And that's all for me for now. Thank you.
Okay, thank you very much. Okay, some questions? Yeah. Okay, thank you very much. Okay. Some questions? Yeah.
Thank you very much. Thanks a lot for a very comprehensive study. I have 2 very quick questions, actually. The first one is I wondered if you considered or had the opportunity to test out taking photos and images of these individuals and then compare that and do the morphometric measures on the computer. Just thinking from a long-term perspective, if that would be possible, then you can train image softwares and that would hopefully make your life easier. And the second quick question was, I was wondering if you considered the life history stage of the individuals as part of, as an effect to the measurements, so if these were mature individuals, immature individuals, and which reproductive stage they were, etc.
Okay, I'll answer first the first part. I have taken many pictures of the samples, each one sample, males and females, but they're very, very tiny differences with the naked eye. That's why I showed you the pelvic fin and not any other differences between females and males. That's why we need, I think, to use morphometry to differentiate them from other species, or within the species, what are the differences, but with statistics, not with pictures. This is very difficult with pictures. For instance, the nostrils or the distance from the snout to the eye. It is very difficult to see it with the eye. That's why I want to.
Might be misunderstanding. I was meaning taking like detailed pictures and then taking exactly the same measurements that you've been taking for the morphometrics, just on a large computer screen.
That will take a great amount of time to do it. I try, but it takes a lot of time to make the measurements with the photos than do it actually on the field or in the lab. It takes me less time to do the morphometry, the measurements with meter and all the other apparatus. You had another question as well? The second question?
Yeah, hi. Thanks so much. It was similar.
You had another question as well? The second question?
Okay. Another question?
Hi. Thanks so much. It was
There.
similar.
Thank you, Ada.
Hi.
I think you said it, but I'm not sure what was the answer. You checked for width to length ratio differences between the males and the females?
Yes, width, total width. Yes.
Width to
Yes, I did that. You mean if there was a difference? Yes. Let me see if they were. Yes. Yes, I did that. In comparison to total length, you mean? Okay,
Yes.
not in the allometry. Yes, there was a difference. Males, I think they have shorter width, width of the.
Because we got this result as well,
Yes,
and
there was
we
a
thought
difference.
maybe it's just because the females are
growing bigger.
They're taller when they lie on the table, so maybe it's changing the
Yes.
the measurements if we do it on the body.
Maybe. I will see what's the reason. As soon as I have bigger males and females from the surveys I'm carrying out in 2019, I will add these results on my results, on these ones, and see why this is happening, why they have differences in width and all the other measurements, the most important at least. Some might be minor. But yes, there are differences, as you can see.
Thank you.
Okay. Okay, thank you. Another more questions?
Great. Thank you
Okay,
very much.
Thank you. Thank you again. Our last speaker before the coffee break is della, distribution and length-weight relationship of 4 deep water shark species from the Antalya Bay, Eastern Mediterranean. Distribution and length-weight relationship of 4 deep water shark species from the Antalya Bay, Eastern Mediterranean.
Hello, everybody. Firstly, I would say that my English is not good. Therefore, if I need, I can speak Italianish to say qua migliore. That my English is not good. Therefore, if I need, I can speak Italianish. The study area is located in Antalya Bay in sub-area 24. In this area, the stock assessment data is very scarce. In our study area, in the last 20 years, the deep water troll fishery has developed to basically red shrimps species. But in these fisheries, high number of elasmobranch species are caught as bycatch and discarded. Therefore, a doctoral study is being conducted on the population dynamic and structure of four deep-water bycatch shark species in a study area in Antalya Bay. And this presentation is a small part of the study, and following informations are shared. The abundance and biomass indices, length-weight relationships, and the bathymetric distribution of the shark species. The survey was performed in September 2016 with the research vessel, and sampling area was divided in five bathymetric strata between 200 and 699 meters. We used a conventional bottom troll with a 44 millimeter cordant mesh and with a 24 millimeter cover mesh. And towing duration was 1 hour, and average speed was about 2.5 knots. 3 holes at each stratum and totally 50 holes. And after for each holes, sharks were sorted by species and was transferred in the laboratory. Sex determination, measurements of total length and total weight, and other analyses were performed in the laboratory. Abundance and biomass indices were estimated by using the ATRIS software. So, during the survey, about 1,200 shark individuals belonging to 4 species from the 4 families were collected. These species are Ethnopterus spinax, Centro granulosus, Squalus blainvillei, and Gallus melastomus. And Ethnopterus spinax was the most abundant shark species with about 600 individuals. And total length of the species ranged from 9.5 to 33 centimeter. Sex ratio was 1:1, and the species distributed deeper than 300 meters, and the highest value of abundance and biomass were obtained in the deep stratum of 500 meters. And second species, Gallus melastomus, more than 600 individuals of species was sampled. Total length of the species ranged from 11.5 to 43.3 centimeter. Also, sex ratio was 1:1. This species also distributed deeper than 300 meters, and the highest value of species of abundance and biomass were obtained the 400 meter of the stratum. And another species, Squalus blainvillei, blainvillei was sampled, and total length was between 20 to 64 centimeter, and sex ratio was also 1:1. And this species distributed deeper than 300 meters, and highest value of abundance and biomass were obtained in the depth stratum of 400 meters. And last species, Centro granulosus, was the less sampled species, and total length ranged from 49 to 96.5 centimeter, and sex ratio was not 1:1, and the species distributed deeper than 400 meter, and highest value of the abundance and biomass were obtained in the depth stratum of 600 meter. In conclusion, will the Gallus melastamus and Squalus plainveli mainly living on the 400 meter stratum, Ethnoperux spinax and Ethnoperux spinax on the 500 meters, and Granulosus on deepest stratum. The smallest specimens are sampled mostly on the methylbathymetric line of upper slope, and larger specimens are along the bentopelagic slope for all 4 shark species. And total length of 4 species had a strongly positive correlation with depth stratum, and the b-value of length-weight relationship ranged between 2.62 and 3.16 for the females, and between 3.04 and 3.56 for the males of 4 species. And we hope that the results of the completed study will contribute to the knowledge of the shark population among the Levanten basin. Thank you very much. Okay.
Okay. Thank you very much. Question?
Okay, Aylin, you are Turkish and you can speak Turkish.
Hi, I was wondering what depths do the
Sorry,
fisheries...
Sorry.
Okay.
Okay.
What depths do the fisheries fish to?
Sorry. Posso parlare italiano? Io ho capito. Also, and the deep water fisheries was between 350 till 650 meters.
Okay.
For the red shrimps.
Okay.
Okay.
Hello, thank you for your presentation. And I was wondering whether you find any differences in sex or size regarding with the bathymetry. Is there more males in any depth, something like that?
Yeah. My presentation is in preliminary results. And the study is, come si dice? Sorry?
Progress?
Yeah, in progress. Then after the study, we can analyze all these results.
Okay, thank you.
Okay. Okay, another question here.
After the study, we can analyze all these results. Okay, thank you.
Hi, thank you for your presentation. I was very impressive amount of sharks. I was wondering how many hauls you did to capture all those sharks, because you were sampling in 1 month in September.
September, yeah.
But how many hauls actually?
Hauls for each stratum, and between 200 and 699, also together 15 hauls.
Wow, that's a very impressive amount of sharks. Do you have any agreement with the fishermen or cooperation so you can check the populations changing over time? Have you tried that?
We will, but we cannot any agreement with fishermen. The fishermen will not work with the scientific.
We'll talk later about
agreement
Hello. Did you get any batoids in your trolls? Did you get any race skates?
Yes. Dipterus oncorhynchus.
Is it part of your study as well, or is someone?
In between, in depth of more than 400 meter of, and Raja Clavata in depth of more than 400.
Is it part of your study, the race, or someone else is analyzing?
Yes, but not in
Not
this
now, but it
presentation,
is.
yeah.
Great. Is it part of your study?
Okay.
Not enough.
Not now, but it is.
I can take
Just another question.
the questions during the
The coffee break.
Yes, I am here. I can take
Another question.
questions during
Yeah,
the
one more question.
Yes.
I'd like you to speculate. Well, do these sharks feed on the shrimp? Do
Poi- Sorry? Si-
I wondered if you could speculate on whether overfishing would affect the sharks directly, or they'll be affected by the loss of their food with the shrimps being overfished. I'm sure the shrimps are being overfished, and the sharks are bycatch. I just wondered if you could speculate about whether the sharks will run out of food or be fished out by being bycatch. It's a difficult question. It's a ridiculous question. So.
Just
Okay, in the coffee break we can
Okay, yeah.
you can continue. Okay, we are.
Thank you very much.
Thank you again. Okay, thank you. We are perfect in time, Emilio. Thank you to all the speakers and to all of you. And we have to come back within 30 minutes. Okay. See you later. And to all of you. And we have to come back within 30 minutes. Okay. See you later.